Welcome students and friends. Today I am discussing about RH alloimmunization, or also called as isoimmunization. It is a little, though it is interesting, it is little bit tricky kind of topic, just like mathematics. If you know the formula, you can answer any questions. Here also, if you know the pathophysiology, you can understand the disease very well and treat it accordingly. So it is very important to know the pathophysiology. Okay, for any disease, for that sake, knowing pathophysiology is very important. So when does this RH incompatibility or RH alloimmunization occurs? It occurs if the mother should be RH negative. The mother is RH negative and she bears a fetus which is RH positive. RH positive fetus, and the mother is RH negative. Then only this RH alloimmunization occurs, leading to its consequences. So what are the consequences? Because of antigen antibody reaction, there is lysis of fetal RBC, leading to hemolysis and anemia and its consequences. So it will lead to hemolysis and anemia in the fetus and its consequences. So we'll learn one by one in the next slides. So what is this RH antigen? The RH antigen, the RH stands for rhesus because it was first discovered in 1940 in rhesus monkey. So the RH comes from this, the RH name comes from this rhesus monkey. And it is the second most important blood group system. Which is the first one? Definitely we know it is the ABO blood group, which is very important one, following that RH typing. So it is said that there are more than 300 plus antigen present on the RBC cell membranes. Amongst them, the ABO blood grouping and RH typing are the very commonest group system which we have to know. So this RH antigen or RH factor is nothing but it is a protein, transmembrane protein. Okay, so suppose if this is the RBC, this is just graphical representation. If this is the RBC, the RH proteins are present on the RBC membrane. Okay, so these are the RH factor, RH antigens. These are nothing but protein present on the RBC membranes. So there are 49 antigens or 49 factors belong to this RH blood group system. Amongst them, 5 are very important. Only 5 we have to know. Amongst that, it is D, C, small c, capital E, and small e. Okay, so easy to remember D, C, and E. Okay, small and capital, except that there is no small d. Fine. Okay, so and amongst these 5 antigens, the D antigen is very important and it is very immunogenic. So whatever we are learning in the further slides, it mainly depends on this D antigen, right? So the gene for this protein is present on chromosome number 1. So suppose if this is chromosome number 1, okay, chromosome number 1. So what happens? The gene encoding for this RH or RH antigens is present on chromosome number 1. So there are 2 closely associated genes, the RHD and RHCE. These are closely associated genes, and these genes are present on chromosome number 1. So they encode for this protein. This RHD encodes for D protein, and this RHCE encodes for this C, small c, capital E, and small e proteins. Understood? So these are the genes. These are the gene present on chromosome number 1, and these are the proteins present on the RBC membrane. Okay. And why we have to know this? Because the incompatibility, as I told, leads to hemolysis. So that is called hemolytic disease in fetal period and in the newborn period or the neonatal period. So RH incompatibility leads to hemolytic disease of fetus and newborn. So we usually, by RH typing means we usually how we express RH typing as either RH positive or RH negative. Suppose if we ask, if we are telling the blood group, we tell A positive, A negative, O positive, O negative, like that. That positive and negative is nothing but RH typing. Fine. This RH typing uh, uh, is reported as either positive or negative. This depends mainly on D antigen. Okay, so we learned in the last slide that there are 5 antigen. D, 5 antigens are there. D, C, E, E. Okay, so we learned there are 5 antigens, but amongst that, the positive and negative depends mainly on the D antigen because it is more immunogenic. So this is considered the most important one. Okay, suppose if there is an RBC, for example, this is a RBC, it contains D antigen on the membrane. So this individual is called as, so this is the D antigen present on RBC cell membrane. So this individual is called as Rh positive. Depending on the ABO group, we tell as either A positive, AB positive, something like that. But these individuals are Rh positive. Suppose if there is another individual, his RBC, his or her RBC doesn't contain this D antigen on the surface. Okay, then this person is told as Rh negative. So Rh positive and Rh negative depends on the presence or absence of D antigen. If D antigen is present on RBC, then the person is called Rh positive. If the D antigen is lacking on the RBC, then we call the person as Rh negative. And very important, the absence of D antigen, the absence of D antigen or D gene is represented as small d. And the prevalence of RHD alloimmunization is 0.5 to 0.9%, as given by Williams Obstetrics. So, and severe hemolytic disease of fetal and newborn is seen with anti-D, anti-KEL, and anti-C. Fine. But here in this presentation, we are mainly learning this D because it is most commonest, more than 90-95% of the cases caused by this D antigen. So we are learning mainly this antigen. So there are other causes also which may cause other causes for hemolytic disease of fetus and newborn, like Lewis antigen, Duffy antigen. They are also other blood group system. They are also involved in hemolytic disease of fetus and newborn, but the major cause is this anti-D. Fine. So hemolytic disease of fetus and newborn is caused by anti-D, anti-KEL, anti-C, Duffy, and Lewis. So these are the various blood group system. I'm not going into the detail. So we can see this Lewis also causes hemolytic disease of fetus and newborn. Even the Duffy can also cause hemolytic disease of fetus and newborn. So coming to a little bit about the history. Okay, so as I told in my first slide that Landsteiner and Wiener discovered this Rh antigen in 1940 in a rhesus monkey. And erythroblastosis fetalis, it is nothing but it is the consequence of Rh incompatibility. Rh incompatibility leads to erythroblastosis fetalis. What is it? I will explain in the further slides. Mainly there will be hydrops, anemia, and hyperbilirubinemia. So erythroblastosis fetalis was first described by Diamond. He described the features of the erythroblastosis fetalis in 1932. And Levin et al. Told that the erythroblastosis fetalis is due to maternal Rh isoimmunization. They told that maternal Rh isoimmunization causes erythroblastosis fetalis. Fine. And anti-D prophylaxis. Okay, anti-D prophylaxis means the antibodies to D antigen were used for prophylaxis to prevent Rh incompatibility, and that was started in the year 1969. So very important to remember, Landsteiner and Wiener identified this Rh antigen in 1940. So these are the classification of Rh system. This Wiener is the old classification and the Fisher race is the new classification, and I am not going into the details of those things. So these are the examples for the gene complex and the antigen. So the gene complex is present on chromosome number 1, and this antigens are present on RBC membrane. Okay, so all the three genes are there. See here. So depending on the genes, there will be the proteins or antigens on the RBC cell membrane. So if you just look at this, just for knowing sake, I'm telling this, all these individuals are Rh positive, isn't it? Why they are as a Rh positive? Because they contain this D protein on the RBC membrane. Okay, all these ones are Rh negative, right? They don't contain this D. Very important, this D, what is this small d? I told there is no small d gene, no small d gene or D protein. So what is this D? This D means it is the absence of capital D gene or the antigen. So the absence is represented as D. Okay, there is no D gene. The representation sake, you put this small d. Fine. So, and the gene for this, once again, I'm telling it is located on chromosome number 1, arm P. Clear? Hope it is clear. So, when we know the gene, we have to know a little bit about the inheritance, very important for the management. So, it is inherited as autosomal dominant. So, the Rh positive individuals can be either homozygous, means suppose if this is chromosome number 1, okay, the both arm will contain D gene. So D gene is present on both the arm, or the P arm of both the chromosome will contain this D gene. Okay, heterozygous means chromosome number 1. Okay, so one arm will contain D gene, it is absent in the D gene is absent in the other arm. Fine. So homozygous, they have two genes on both the arms of the chromosome. Here in heterozygous, one arm will have, the other arm it is absent. That is for representation, we put it as small d. Fine. So heterozygous. Then Rh negative individuals. Rh negative individual means this is the chromosome number 1. So there is absence of D gene in both the arm of chromosomes. So there are no D genes. So that's why it is represented as small d and D. Fine. Is it clear? So suppose we'll take a scenario where mother is Rh negative. Mother is Rh negative. Okay. The father is Rh positive. Rh positive can be either homozygous or it can be heterozygous. Suppose if mother is Rh negative and father is Rh positive, so the offsprings can be, if you combine and write like this, D and D. Okay. So if you combine this and this, so what is the blood group of the fetus or the Rh typing of the fetus or the baby? The babies will be, all the babies will be Rh positive. Okay, 100%. Okay, so all the babies are Rh positive. Suppose if you see the heterozygous, so if you match and write capital and small d because both are small. Okay, you combine and write like this. So what are these individuals? So these babies are Rh negative, whereas these individuals are Rh positive. Though they are heterozygous, but still they are Rh positive. Here also they are heterozygous, but still they are Rh positive. So 50-50 chance. So 50% Rh negative, the babies are 50% Rh positive, 50% Rh negative. Okay, so why we have to know this? Because I told in my first slide that Rh negative mother, if she has Rh positive fetus, it leads to Rh isoimmunization. Okay, so herein, if the father is heterozygous, so the risk is reduced. Okay, so the risk is reduced by 50% because there is 50% chance of baby being Rh negative. So then if the mother is also Rh negative, baby is also Rh negative, there is no need for monitoring, or there is no risk of, or we can tell there is no risk of isoimmunization in these cases. 50% definitely it is there. The other 50% there is no risk. Fine. What happens if the mother is Rh negative mother, and the father is father is also Rh negative. So the resultant fetus will be 100% they are Rh negative. Okay. So there is here also there is no risk. Here also there is no risk. Okay, in heterozygous 50% there is no risk because 50% chance of baby being Rh negative. Here also, if the both mother and father are Rh negative, then also there is no risk of isoimmunization. So that's why the summary of this is very important. If any patient with Rh negative pregnancy come, first we have to see for the paternal Rh status of the father. Okay, suppose if it is negative, there is no risk. Okay, no need to worry. And suppose if he is positive, then we have to see for the zygosity. Then we have to see for the zygosity, whether he is homozygous or he is heterozygous. Clear? So coming to the incidence of Rh alloimmunization. After 2 births, if she has not taken any prophylaxis, then the risk is around 13 to 16%. If we give only postnatal anti-D, the risk, see the reduction in risk from 16%, it has reduced to less than 2%. And if we give anti-D at 28 weeks, that is called routine antenatal anti-D prophylaxis. Routine antenatal anti-D prophylaxis means we give anti-D, that is antibody to D antigen. If we give at 28 weeks combined with postnatal anti-D, the risk is reduced further to 0.2%. See, from 16%, the risk has come to 0.2% with the administration of postnatal anti-D and anti-D at 28 weeks. So just to break the monotony of the talk, so we'll answer a few questions. So mother and fetus Rh incompatibility can occur if the mother is, repeated many times, the mother should be Rh negative. Pregnant mother should be Rh negative and her fetus should be, incompatibility means what? Baby should be Rh positive, then only isoimmunization occurs. And which of the following sentences is true? So we have to find the true sentence for the Rh positive people. So Rh positive people. So we'll come from here. It is homozygous recessive. No, it is homozygous and heterozygous, but it is not recessive. The D antigen is autosomal dominant. Okay, so this statement is wrong. And all are homozygous. No, there is also heterozygous and it is not recessive. So this sentence is also wrong. And B, the homozygous autosomal dominant. Yeah, it is autosomal dominant, but it is all Rh positive people are not homozygous. Okay, so this is also wrong. So what is the, so here the last sentence is autosomal dominant. Yes, they are all autosomal dominant. D gene is autosomal dominant. They can be either homozygous, right? They can be either heterozygous. So this is the right answer. So Rh positive individual, the gene is autosomal dominant and it is inherited, or sorry, not inherited. The zygosity can be either homozygous or heterozygous. Fine. So coming to the pathophysiology of the Rh alloimmunization. So before going to the pathophysiology, let us learn some definitions. So I have been telling what is, I've been telling a number of times, alloimmunization, isoimmunization. You must be wondering what is this. Immunization means there is production of antibodies, right? So there is production of antibodies. The antibodies are produced against antigen. So antigen is also there, and this antigen is derived from the same species individual. That's why the name allo or isoimmunization. Allo or isoimmunization means the antigen is from the same species individual. Okay, for just knowing sake, I'm telling what is autoimmunity. Autoimmunity means there also antibodies are produced. Those antibodies are produced to against our own self antigens. Means my own self antigens. If I produce antibody, that is called autoimmunity. Autoimmunity we see in HIV and all, no? So there the antibodies are produced to the body's own antigen. Here it is to the antigen of the individual from the same species. That's why it is called allo or isoimmunization. Here, who is producing the antibodies in Rh incompatibility? Who is producing antibodies? Yes, definitely. Mother is producing antibodies against the antigen. Where is the antigen? The antigen is in the fetus. Which is the antigen? It is the D antigen. RH antigen in that D antigen. Okay. And this antigen this antigen or the D antigen, the mother lacks this D antigen, right? Then only allo or isoimmunization is possible. Okay, so it is production. The mother is producing antibodies against the fetal antigen which she lacks. Means she lacks this Rh antigen, right? Because she is Rh negative. So erythroblastosis fetalis. It is characterized by hydrops fetalis, severe anemia, and icterus gravis neonatal. And here blast cells are produced into the or pushed into the circulation. What do we mean by erythroblast? Erythroblast means premature RBCs are pushed into circulation. Why they are pushed? Because there is hemolysis is there. Hemolysis leading to anemia. Fine, anemia is there. Therefore, the oxygen supply to the periphery is reduced. Therefore, these premature RBCs are pushed into the circulation. Whose circulation? Fetal circulation. So erythroblastosis fetalis means there is hydrops fetalis. What is hydrops? I will be explaining. Severe anemia will be there, and icterus gravis. Icterus is because of hemolysis leads to increased bilirubin. So all these features included, it is called as erythroblastosis fetalis. What is hydrops fetalis? In hydrops, there is abnormal accumulation of fluid in the third space, more than one area. So where all the fluid get accumulated in as ascites, fetal ascites, pleural effusion, pericardial effusion, skin edema. If it is more pronounced around scalp, it is called scalp edema. So these are the area where the fluid gets accumulated. So if two areas, means if there is ascites and skin edema, then we tell the patient is having hydrops fetalis. Or pleural effusion with ascites also. If mainly the idea is more than one area, means two areas should be involved, two or above. Fine. So this is hydrops fetalis. And very important thing you have to know about fetal-maternal hemorrhage. So what is fetal-maternal hemorrhage? As the name suggests, the fetal cells are seen in maternal circulation. Fetal RBCs get into the maternal circulation. If you see the placental anatomy, so there will be chorionic villi like this, and the space in between the chorionic villi is the intervillous space, and these are a villi, chorionic villi. Where will be the fetal blood vessels? The fetal blood vessels run in this villi, right? It will be in the villi. So the fetal blood will be in this chorionic villi, circulating in the fetal blood vessels, whereas the maternal blood will be in the intervillous space. So there is no direct contact between maternal blood and the fetal blood. It is through this barrier, placental barrier, or the interface, feto-maternal interface, through which the exchange of tissue occurs. What happens? Because of small interruption in this, some amount of fetal RBCs gets into the maternal circulation. That is called feto-maternal hemorrhage. So, fetal cells in maternal circulation is called feto-maternal hemorrhage. So coming to pathophysiology proper, so first pregnancy, very important to remember, it is usually more than 95% or roughly 99% of the times, usually the fetus is unaffected in the first pregnancy. Okay, so here we'll see the mother is Rh negative. She doesn't contain, that means she doesn't contain D antigen or any D antibody. She is Rh negative mother. Now she's pregnant with Rh positive fetus. The fetus is Rh positive means there is D antigen on the fetal RBC. So there is D antigen on the fetal RBCs, right? So baby is Rh positive. Now what happens? There is feto-maternal hemorrhage is there. Sorry, feto-maternal hemorrhage is there. So the RBCs are pushed into maternal circulation. So there is primary immune response. So the primary immune response will be there against this D antigen because it is a foreign protein. So definitely the immune system recognizes it as antigen, and there is production of antibodies. As you know, the primary response, IgM is produced. Okay, as primary response, IgM is produced. It is a large molecular weight. It is of large molecular weight and it is pentamer, so it doesn't cross placenta and go to the fetus. So since it has molecular weight, it is a large molecular weight, it does not cross placenta. Fine. This is primary immune response. So what happens later on? There is secondary immune response. Okay. Secondary immune response, there is production of IgG antibodies, which I'll be explaining in the further slides. Okay, so for the production of IgG antibodies, it usually takes 3 to 6 months. Okay, by that time, the pregnancy, the fetus would have, that means the fetus would have delivered by that time. Okay, so the time is 3 to 6 months. By the time, usually the feto-maternal hemorrhage occurs around labor. Okay, more than 90% of the times it occurs during labor. Fine. Okay, by the time she produces IgG immunoglobulins, she would have delivered. Fine. Okay, so there are few exceptions for this rule. This rule means I told no, first pregnancy it is unaffected. Exception means first pregnancy the fetus get affected. Fetus is affected in first pregnancy only. Sorry for the bad handwriting. Okay, fine. So what are those? Means what I mean to tell is she has been sensitized before pregnancy only. Means she has this anti-D antibodies already. She has this prepared antibodies already. So when is this possible? Either she has received mismatched blood transfusion. In the sense she is Rh negative, she must have received Rh positive blood in previously. So this is very rare. Okay, in medical practice, we don't do this mismatched blood transfusion. Suppose in case or in a very dire life-saving procedure, something, a scenario like that, dire emergency, where Rh negative blood is not there, if they have transfused, of course, this is very, very, very rare phenomenon. She has received Rh positive blood. In that case, she has anti-D antibody already. So her first pregnancy is likely to be affected. Okay. And the second one is grandmother theory. I'll be explaining. The third one is antenatal sensitization. Means very early in pregnancy. Very, very early in pregnancy, if she's sensitized maybe because of threatened abortion or could be due to she has undergone chorionic villus sampling, something, and not received prophylaxis. A very rare scenario like that, where she has got sensitized very early in pregnancy and she has time to produce IgG. So her first pregnancy is likely to be affected. Okay, this is also very rare. Okay, so let us see what is this interesting theory, grandmother theory. So this is the current mother. She is Rh negative and she bears a fetus which is Rh positive. Fine. So current mother during her in utero period, when she was fetus, during her in utero period, she was exposed to the Rh positive cells of her mother. Okay. So if the grandmother's Rh positive RBCs during this mother's in utero period, if these RBCs have entered her circulation when she was in the fetal period, okay, so she has also produced anti-D. Means the current mother has produced anti-D during her fetal or early neonatal life. So she has preformed anti-D now. So likely that her first pregnancy, she might affect her fetus. First pregnancy, she might affect her fetus because of the presence of anti-D antibodies in her circulation. Fine. The current mother during her neonatal life, the current mother during her fetal life, if she was exposed to her mother's Rh positive cells, so antibodies are formed already. So likely she is going to affect her fetus, the current fetus in her first pregnancy only. So these are the few exceptions where the first pregnancy, the fetus is affected. And all these phenomena, I'm telling repeatedly, it is very, very rare. Okay. It's very, very rare. So usually more than 19 to 95 to 99% of the times, the first pregnancy is unaffected. So what happens in the subsequent pregnancy? I told first pregnancy it is not affected. So what happens in the subsequent pregnancy? So now the mother has been sensitized, means she was exposed to D antigen. She has produced IgM antibodies. Now she is just not Rh negative mother. She is already sensitized to the Rh antigen. So she has anti-D antibodies and now she bears Rh positive fetus. Okay, so the Rh positive fetus will have D antigen, fetal-maternal hemorrhage will be there. So the D antigen or the fetal RBCs containing D antigen enter the maternal circulation. So this is the secondary exposure. So secondary exposure, there will be amnestic reaction, means there is increased production because memory will be stimulated. Memory of the immune system will be stimulated. So what happens? There is increased production of immunoglobulins. So which immunoglobulin is produced? It is the IgG. Now we have the secondary response. We got increased production of immunoglobulin, and that is because, and that also the immunoglobulin is IgG. So amnestic because there is memory of the immune system against this antigen, there is increased production of IgG immunoglobulin. So this is called amnestic reaction. These IgG antibodies are low molecular weight. So definitely the placenta is not a complete barrier. So this low molecular weight IgG cross the placenta and comes to the fetus. So what happens in fetus? So there is antigen, the anti-D crosses placenta and it binds to D antigen. So what happens? There is sequestration of this fetal RBCs. Antigen antibody reaction occurs and this are taken up by the spleen, that is reticular endothelial system, and there is sequestration of fetal RBCs leading to hemolysis, thereby leading to anemia. Fine. So here there is production, the second exposure, memory is there. So secondary exposure, increased amounts of IgG are produced that cross placenta and binds to the D antigen. So there is sequestration of this fetal RBCs and the resultant hemolysis and anemia will be there. So what are the problems with hemolysis?